Okay, I assume you've learned the basics of genetics and you've been practicing some problems. I just want to give you some additional examples of different things I see people make mistakes on, and hopefully that will prevent you from doing the same thing. So I'm just going to start with a kind of generic problem, but looking at 3 different genes. So I'm not going to tell you any phenotypes because you'll see that doesn't matter. This is the cross I'm having you look at. Okay. And remember, if you're using notations of, like, for example, the C here, where the big and the little look similar, you need to put the line over it, but obviously something like the B or the A is quite different, so you don't have to show that. Okay, so we have these 2 organisms crossing, doesn't matter what they are. And my question to you is, what's the probability of the offspring having the dad's phenotype? Okay, so you're thinking, oh my gosh, she didn't even tell me any phenotypes. How am I supposed to know what A is or B? I can't even answer this question. But if you just think about it, if I haven't told you any differently, this is a complete dominance scenario. So if we think about how would I look like dad, and I do have to tell you which one is the mother and the father, so this will be the male, so that's the dad, and this will be the female, that's the mom. So we're going to ask, what's the probability of the baby having a phenotype like the dad? So all that matters is that we know if it's going to be a dominant or recessive phenotype. So dad has one big A and one little a, so that means that to get the phenotype of dad, we have to have one big A, at least. Dad has 2 big B's, so to get the phenotype of dad, we need at least one big B to have the dominant phenotype. And then dad has 1 big C and 1 little c. To get the phenotype of dad, we need at least 1 big C, because it's the dominant phenotype. So indeed, the dad has the dominant A, B, and C phenotypes in this case. Remember, dominant doesn't mean better or more common or anything else. It just means that when you have, you only need 1 copy of that version of the gene or allele to show the phenotype. So what you're going to do is to solve 3 separate crosses, or in Punnett square form. So the A's crossed, the B's crossed, and the C's crossed, and that's because these are, I have not told you these are linked genes. They're genes on separate chromosomes, so you can treat them all as separate entities because they do their own thing. And then we'll just have to multiply the probabilities together at the end, because obviously this would be very it would be way too big and take so much time to try to do it in one big Punnett square. So it's a waste of your time. Don't do that. Okay, so let's just take the A's. So we have the cross actually big A, little a, cross with little a, little a. Now if we think about this, what does this mean? This means that dad has 2 types of sperm. Half his sperm carry a big A allele, and half his sperm carry the little a allele. There's 2 versions. Mom only has 1 version of eggs. All of them carry the little a. So we only have to write that once. Don't waste your time writing it twice, because that's the same exact thing 2 times. So all we have is 1 egg type, and if that egg and this sperm cross, we get big A, little a. If this egg, same type of egg, and this sperm cross, we get little a, little a. Okay, so that's our A's. Now our B's, we have big B, and big B, little b. So we'll just think of those as a separate cross for now. Okay, so in this case, dad only has 1 type of sperm, big B. Mom has 2 types of eggs. Half of them are big B, half have a little b, big B, little b, excuse me, 2 big B's and then big B, little b. So those are the 2 types of B combos you can get. And then the C's are here, big C, little c, big C, little c. So that's a classic monohybrid cross. You should kind of know that by this point, but I'll do it anyway. So dad has 2 types of sperm, a big C sperm type and a little c sperm type. Mom also has 2 different types of eggs. You see why I have to write the lines over it, because, you know, I can't tell if it's a big or small c just by what it's written as. So this is the 1 to 2 to 1 or 3 to 1 phenotype. Great. So those are our 3 crosses. Now we just need to say what do we need to happen in order to get our goal. So then we say, what's the probability of getting at least 1 big A? Well, only 1 of 2 of our boxes say that. times, and that's because we need both these things to happen. So times the probability of getting at least one big B, well, both of our boxes have that, 2 out of 2, or that's just 1, times the probability of getting at least one big C, that's 3 of our boxes out of 4, 3 out of 4, and then we multiply that, so you just multiply across the top, and you multiply across the bottom, and then that tells you the probability that the baby is going to be the same phenotype as the father, 3/8. And just leave it as a fraction, don't put it in percentage. So that's going to be probability. So this is the same cross, but what if I asked you what the probability of having a triple heterozygote, big A little a, big B little b, big C little c child? So you can pause and try to solve that. We already have the Punnett squares done, so we can just look at those. All right, so what's the probability of getting a big A little a? Well, that's 1 out of 2. Times big B little b, 1 out of 2. Times big C little t, c. That's 2 out of 4, or 1 out of 2, which equals 2 times 2 is 4, 4 times 2 is 8, so there's a 1/8 chance of having a child that has the 3 heterozygous alleles, or that's heterozygous at the 3 loci, for the 3 genes. What if we have this cross? Big A little a cross with big A little a, monohybrid cross. And I'll tell you that the big A genotype, remember genotype is which alleles it has, that is lethal. And I'll also tell you that this is a dominant trait. So my question is, what's the probability of a kid with the trait? So you can pause and see if you can figure that out. Okay, so you might know this by now, classic monohybrid cross, but I'll do it again. There's 2 types of eggs and 2 types of sperm for both the mother and the father. So we have here probability of kid with the trait. Well, the thing to note that's different here is that we said that this one's lethal. So we never see any offspring with the big A genotype. That one's dead. So we have to take that out of the Punnett square because we might get fertilizations like that, but we'll never get offspring like that. So so that option won't be part of the probability or the fractions that we see. Okay, so of those left, probability of a kid with the trait, so it's a dominant trait, so the ones with the trait have a dominant allele, at least one, so that's these 2. So the probability of a child with the trait is 2/3, instead of what you might think of as 1/2. So this is when you see things that are different, maybe with 2/3, 1/3, something like that. It might suggest something lethal, or that you're not counting a group already. So for example, I could ask this question in a similar way, but instead of saying lethal, I do something like, let's just do the same old. Not everybody's heterozygous for everything, but okay. So you have a kid with a disease. Let's say it's homozygous recessive. So you know that the child that has a homozygous recessive, has the disease, has this. You have a second child that's healthy. Okay, you have a kid that's healthy, and you want to know what's the probability that that healthy child is a carrier now that you found out that you have this allele in your family. So then you have to take into account the kid is healthy already, which means you know it can't be this genotype, which means you're only left with these 3. So the probability of the kid being a carrier is these 2. And so that, again, is a 2 out of 3 probability that the healthy child that you have already is a carrier. Unlike if I ask you, an unborn child that you don't have yet, what's the probability that your next kid, let's say you have this kid with the disease, what's the probability that the next kid is a carrier? In that case, you don't know which of these 4 the kid is going to be. It could be affected. So you have all 4 options still. So if I asked you what's the probability of the next child that you don't know being a carrier, that would again be the 2 out of 4 or 1/2. So that's the probability of the next child versus the probability of the healthy child. This is the same thing with males and females as well. Here's an example. This is going to be about clotting factor alleles, so this would be what could lead to something like hemophilia. All right, so you have 2 versions of this H gene. So there's the big H, the dominant version of the gene, the allele that codes for a functional clotting factor. And then there's a different version of the gene, which just means a different DNA sequence, which codes for no clotting factor, meaning probably it has like a nonsense mutation where the protein doesn't get folded because the polypeptide's too short. And so the clotting factor doesn't work. And just to remind you, we're also including Y here because this is on the X chromosome, the H gene. But the Y doesn't have the H gene. I'm just putting it in there to remind you that you're doing X's and Y's for males and females. And so I'm going to ask you, if you have a carrier mom and a normal dad, what's the probability that your next child has hemophilia? Or just could say your kid has hemophilia. I don't give you any more information than that. Okay, so you can try to solve this. I'll give you a moment to pause. Okay, what I like to start out with is just making sure I don't make a silly mistake and forget my parents. So there's XX and XY, so the mother and the father. I tell you the mother is a carrier. Carrier automatically means A, it has to be a recessive trait you're looking at. Can't carry a dominant, because if you have a dominant, you show it. The carrier mother, though, is going to have not show it, so she's going to be phenotypically normal, but she carries that recessive mutant allele in this case. And then the father only has, we know what he is because he only has the H gene on his X, and he's normal, so he must have a single copy, call that hemizygous for the H, and he has the big H version. Okay, so if we cross those, just like normal, but you have to just use your X's and Y's. So here's your dad, here's your mom, big HY, little HY, big H, little h. Okay, so now if my question, you have to pay attention, is asking you what's the probability a kid has hemophilia, that can be anybody in this pedigree. So the only one that manifests the hemophilia, which has hemophilia, is this individual here. So the probability that your kid or your next kid has hemophilia is 1 out of 4. Now on the other hand, if I asked you what's the probability your baby girl is going to have hemophilia. And remember, this does not mean have the hemophilia allele. This means manifest the phenotype of hemophilia, which means you have to have no clotting factor present. So what's the probability of a girl having hemophilia, like you know you're going to have a baby girl? Well, you look here, these are the only 2 girl options, and both of them have at least 1 big H, which means they both produce enough clotting factor to not have hemophilia. So in this case, there's no chance, 0%, no chance that the girl has hemophilia. On the other hand, if I asked you about a boy, let's say you know that you have a boy, you're having a boy, and I asked you what's the probability that your baby boy is going to have hemophilia, these are the only 2 boy options. This one we said is manifesting the hemophilia. This one's top one is not. So that's a 1 out of 2 chance that your baby boy is going to have hemophilia if you know that this is your carrier female and the male is normal. So you can see, depending on what we ask and what information you know, the probabilities can be different. I could even do something like this. And just, you know, these parents are the same parents, and I could say, what's the probability that the kid has hemophilia? But then in this case, you know that it's a square, which you know means that's a boy child. So then you just go and do the same thing as the boy with hemophilia. So if I showed you a pedigree, you have some more information. If you don't know the sex of the offspring, then you do a diamond. So if you saw that, that would mean we don't know whether it's a male or female. And so you'd have to just go back to this probability the kid has hemophilia 1 in 4. But if I were to give you a square or circle, then you know already the sex of the child. So that tells you something there too. Or if I have something where it's like colored in, and I ask you something about like what's the probability that this male is X little hy, something like that. And you had his parents too or something along those lines, and I said, what's the probability this male has X little hy? Well, you'd be like, well, it's colored in, so he has the trait of hemophilia, so it's 100%, or yeah, so it's 100% probability that he has that. So that can be the case if I give you more information. All right, we're going to go and do two separate things on this one, just looking at the same time. So one thing we're looking at in this question is going to be what we call epistasis, and one thing we're looking at in this question is incomplete dominance. And you've seen examples of these before, but I'm just going to make another one, putting them together. So there is a gene, I'm calling it T, that codes for I'll just tell you that it's coding for a protein that's a transcription factor, which means it turns on genes, binds those regulatory regions you learned about in gene expression, and it's going to turn on other genes that are important for making a tail. So when you have the version that we're calling big T because it's dominant, it makes turns on the transcription factor, which turns on the other genes that make a tail during embryonic development of these dragons. So you get a tail. If you have two copies of this other version of the gene, the little t allele, then it codes for a transcription factor that doesn't work, and so you don't get the other genes turned on, so the dragon does not make a tail at all in embryonic development. So it comes out with no tail. On top of that, all dragons have a different gene that codes for tail shape. Even if they don't have a tail, they still have this tail shape gene. It's going to be called gene A. Okay, and there's two versions of this, all right? There's an AS version and an AC version, but I have to tell you that there's actually three different phenotypes. There's a straight tail phenotype, a kinked tail, and a curly tail phenotype. So it turns out that this is incomplete dominance. So when you have two copies of the AS, it gets a straight tail. When you have two copies of the AC, it gets a completely curly tail. But when you have one copy of the AS and one copy of the AC, it's just kinked tail. And so I actually, when I'm doing incomplete dominance, first of all, I don't like to write a big A, little a's, because then you'll forget that there's this kinked version here, and it's not actually dominant recessive. These are both, they're incomplete dominant because neither one shows itself over, quote, the other one. So I actually like to write out the full genotype of the three different phenotypes, and then I just don't make a silly mistake by forgetting that there's this kinked genotype, phenotype in there. So this is what I actually like to do. So that's for the tails. All right, so what we're going to have now, I want you to tell me what the phenotypic ratio of the offspring will be for a kinked tail male, that male has a no-tail father, cross with a no-tail female. She had or has two straight-tail parents. So between these two, what's the phenotypic ratio of the offspring? So you can pause that and see if you can figure it out. Okay, so the first thing you're going to have to do is to figure out the genotypes, obviously, of the parents. So what I'm going to do is write 4 blanks to remind myself that I have 2 genes and there's 2 alleles of each. So I will need 2 different or the same 2 T's, and I'll need 2 A's in order to have my entire genotype so I don't, make a mistake and do that incorrectly. And then I'll do it for the female as well. Okay, so for the male, what am I going to know? So that I just do what I know from the phenotype first off. So he has a kinked tail. That's one thing. First of all, he has a tail. Let's just go with that. He has a tail. So if he has a tail, that means he must have at least one copy of the big T allele, because that gives you the ability to grow a tail as an embryo. And then we can say on top of that, that tail is kinked, which we go over here and we wrote down what kinked means already, so we didn't make a silly mistake with that. So we know it's the heterozygous ASAC. Great. From that, that's all we can tell just from his phenotype. So we'll come back to his parent in a minute. All right, looking at the female, what do we know? She has no tail. So the way to get no tail is to have two little t's. So we can write that in. And we don't know anything just from looking at her about her A genes because she doesn't show those. That's the whole thing here about epistasis, is that the tail, no tail is epistatic to the tail shape. So if you have no tail, you still have a gene for tail shape, and you still have two alleles for that. But you can't see them because you didn't grow a tail at all. So maybe it would have been a curly tail because you have curly tail genes, but if you don't grow a tail, then you don't see that phenotype. So how are we going to figure this out? Well, I give you some more information. Let's go back to the male. We're only missing one of the alleles for him. He has a no-tail father. So what does that tell you? Okay, so you know the father has no tail, which tells you he has little t, little t. If you know the father has two little t's, then he must have given his son one of those. So you know the second allele for that male has to be a little t from his dad. Going on to the future mother here that we're trying to cross, she has two straight tail parents. Okay, so what does that tell you? To get a straight tail, you have to have AS, and both of them are ASAS, which means all the eggs and sperm all have just ASs. That's the only allele that's present for the A gene. So that means that she must have two as well, because both her parents are ASAS, so there's only ASs, so therefore that's what she has. So now we know the genotypes of our two parents we're going to cross. Then we can figure out the genotypes of the children, or the phenotypes, I should say, of the children. Now, one thing I want to point out is that even though I'm going to work these in two separate Punnett squares, the T's and the A's, that you do need to understand what the gametes have in them. So let's say I'm taking this mother here, and I ask you on the exam or something to tell me an egg genotype. So first of all, that means you're writing letters, the different versions of the genes. Okay, so if we think about an egg, remember it's formed through meiosis in animals, and so what that means is that it's going to have only one copy of each gene. So it's going to have one T. Now they're only little t's, that's all there is. So we know that for sure her egg has a little t on the chromosome that the T gene is on. And then she actually only has one version of A, so she has a AS. So we know that her eggs all have the AS. So every single egg actually in this mother would be exactly the same. So if you were doing a Punnett square and you were doing you wouldn't have to do a 16 square Punnett square because you only have one egg. There's only one. So all you need to do is use that. However, I'm going to suggest that you do this not with that kind of Punnett square, but with two separate ones like I like to do, and then probabilities. But if I ask you to write an egg genotype, you're only going to have one of each letter. Same with the male, or for the sperm genotype, you'd only have one of each letter because you only have one copy of every gene because of meiosis. Well, let's just do an example of a sperm possibility, just so you understand. Okay, if I asked you for a sperm possible genotype, you'd say whichever T's you have. So you have either option. So you could say, okay, big T. And then you have either option for the A's. So the male has both options. So perhaps this one has an AC in it. So that's a sperm genotype. So any combination of an A and a T that's present in the genotype of the parent. So this is the parent. Adults always have two of each because they're diploid. But these are haploid. Sperm are and eggs are. So they'll only have one of each type of gene. All right, so I've rewritten the parents here, so it's easier to do a cross on the next empty page. All right, so we'll do them separately. So this is the T's crossed together. So you have two options of sperm, big T and little t. You only have one option of eggs, little t. So this is one of those test crosses. And then for your A's, you have two options of sperm, the AS sperm and the AC sperm. You have one option for eggs. All the eggs have the AS allele. That's it. So you only have two different combos of A's that you can get in the offspring. Okay, AS-AS and AS-AC. Great, so now we want to look at our genotypes. So instead of writing out, or phenotypes, so there's, straight tail, kink tail, curly tail, and no tail. And we could go through all of those, but what I think I'll do is just to look at the phenotypes we see here. Okay, so let's just look at the tail shapes. What are the possible tail shapes? So we have an AS-AS, and actually from before in our other slide we had that written down, so that's straight tail. So it's actually one box out of two, or half, have a straight tail phenotype. And then the other option here, the other box, 1 out of 2, has this combo, which is the kinked tail phenotype. Okay, so we know we have straight tail possibility, kinked tail possibility, and then, so we don't have any curly tail, so we don't have to worry about that genotype. And then we have tails and no tails, so it's possible to have a no tail. So if we look at our T's, we have a big T here, so half the boxes have a tail. Genotype, and then here we have two little t's, which would give you no tail, or tailless, I suppose we could say. So those are our options here. Now what I like to do is to then figure out my genotypes I need for the different phenotypes. So one phenotype that looks like a possibility would be having no tail. Another one would be a straight tail. Another one is a kink tail. I'm not going to bother doing curly because we know we can't get that because we don't have the A's. So I'm going to put 4 blanks down again so I don't get confused. So I know I need A's and T's. All right, so to get no tail, we have 2 little t's and any A's. To get either type, any tail, we have to have a big T, at least one. And then to get straight tails, our A's have to be the AS's, and to get a kinked, it's an A with the AC, it's the heterozygote. And then what I do is I just figure out the probability of each of these parts. So what's the probability of getting 2 little t's? Well, we already wrote it down, but we have 1 box out of 2, so that's, I'm going to write this. All right, they're a little more spread out. Good, so the probability of 2 little t's is 1 out of 2, 1/2. And then anything else goes, so that's just 1, times 1 if you want to think like that. And so in the end, you just have 1/2 probability of getting no tail. So that does not mean necessarily that half the babies will have no tail. It just means that the probability is a half, and then that if you had enough babies, you'd expect about half to have no tail. To get a straight tail, what's the probability of having at least 1 big T? That's 1 out of 2 boxes times the probability of having 2 S's, or AS's, which is another half, which gives you 1/4. And then the probability of having a big T again is 1/2 times the probability of having an ASAC, which is 1/2, which is 1/4 again. Now I've asked you about the genotypic ratio, which is not fractions, it is whole numbers, and you have to make them over the same denominator. So we'll have to make the 1/2 go to 2/4 because it all has to be the same. So just to remind you, 2/4. Okay, so our phenotypic ratio is we expect 2 no tails or tailless to every 1 straight-tailed offspring to every 1 kinked-tailed offspring. And that would be our phenotypic ratio. Also just note that we don't always have to use 4 square Punnett squares or 16 squares or anything like that. You just use whatever is reasonable for the cross that you're given, because not all crosses are heterozygote cross with a heterozygote. So this is an example of epistasis, meaning that the no tail is epistatic to any tail shape. And then this is also incomplete dominance because you have this middle phenotype for the heterozygous genotype. thinking about the dragon tails again, I could ask you something like this. So if you have a straight-tailed dragon crossed with a curly-tailed dragon, what are the phenotypic and genotypic ratios that you expect? So you'd have to remember that, you'd have your alleles written out. The straight crossed with the curly, and you might be able to see this right away, but if you can't, in the straight parent, there's only straight tail alleles, so you only have AS as an option for its gametes. And you only have ACs as an option for the gametes of the other parent, so all you have is AS and AC. That would be every single one of them. So, what is my genotypic ratio? Well, 1 means nothing to me. You can't say 1 AS AC. That doesn't even make sense. So what you'd write here is that they'd all be ASAC, and that's your genotypic ratio. And your phenotypic ratio is all, in this case, kink tails. Now I want to give a simple example of, it's simple genetics, but I want to connect what we mean by like big C, little c, to actual physical things like proteins. So, genes do nothing, right? Genes just code for information. DNA itself does pretty much nothing except for hold the information. It's the proteins or the regulatory RNAs in the end that do stuff. So here is a type of phenotype in pigeons. This is called crest phenotype. So most pigeons you've seen probably look like this with their feathers going down towards their tail. But there are some pigeons that have varying sizes of these things called crests. So their feathers actually grow upward. And there are other genes that modify how big a crest is, but the crest, let's see if you can see bigger ones. Here we go. So, there's other modifying genes for how intense and big that crest is. You can see here's a small one versus really, really extreme crests. But the thing to make the feathers grow the wrong way is a crest, we'll call it a crest gene. And it's just inherited in a complete dominant manner, so nothing fancy there. So we can call that gene for the original version of the gene, or the wild type version, no crest. It leads to the hairs growing down towards the tail, and the recessive version of the gene is the one that makes the hair grow, or the feathers, I should say, grow up towards the crown of the head. So we actually know that the C genes are actually representing a real gene called FB2. FB2, Fs are all types of receptors. They're involved in neuron signaling as well, but these are involved in feather patterns, feather direction growth, it seems. And actually that makes sense because Fs and ephrins, they're involved in where neurons grow and they don't grow. So it makes sense that they would be involved in the pathways, which feathers, directions, and stuff that they grow. So there's 2 different alleles that we're looking at here, versions of the FB2 gene. One we're representing with the big C and the other with the little c. And actually they know what's going on there, that the version of the gene that we're designating big C because it's dominant has an arginine amino acid. In that same spot in the recessive version of the gene, it has actually been mutated and now codes for cysteine amino acid in the polypeptide. When it has the arginine amino acid there, the polypeptide folds into a protein that, like I mentioned, is a receptor. So it ends up with a receptor protein, and it has a shape that can transduce the signal. Remember, transduction from our cell signaling means taking, basically transmitting the signal after the ligand binds into the cell. I'll show you a picture. So it's a signal for feather growth. Okay, and they know this. The cysteine in that shape, in the polypeptide, actually causes the protein to misfold into a misshaped protein receptor. It cannot transduce the signal. And so you don't get the cellular response part of the signaling pathway there, because the cell does not respond to that signal anymore. When there are heterozygous pigeons, so no crest and crest, so let's say you have, from a parent that is what we call true breeding, right? So true breeding means homozygous. True breeding, no crest, true breeding, crest, so you know that you have a heterozygous offspring. What they find is that they have no crest, which is why we call that the dominant allele. That's all it means, is that when you have one copy, you show it. And so what that means is that when their cells during development have, draw a better receptor. Okay, so the cell during development has these receptor proteins on it, and this is where it binds. This is on the cell membrane. And to have the proper binding, make it bigger. Okay, you have to have this type of shape, and then the signal gets transduced here. But some of the proteins in the heterozygote have this incorrect shape down there, so they can bind okay, it looks like, but they don't transduce the signal. So in a bird with all functional receptors, so every protein that's produced from both alleles, because it's homozygous for no crest, gives the signal transduction, which makes the feathers grow downward, so they don't have the crest. In a bird with two copies of this, what we're calling a crest allele, which just means the FB2 version that has the cysteine, has all misshapen receptors, so every receptor is like this, so there's no signal at all, and the feathers grow up. In the heterozygote, and this is where scientists learn whether it's recessive or dominant, half of the receptors are going to be the correct shape and bind the signal and transduce it, and half the receptors are going to have that misshapen and not transduce the signal. So it turns out in this case that half the signal amount is enough to get the feathers to grow in the correct direction, so you don't get the crest. There are cases when you need two copies in order to get enough signal to get a phenotype, and then the other one, then it would no longer be dominant. So in this case, because you only need one copy to get enough of the protein to give the phenotype of no crest, that one is what we call the dominant allele. So, two misshapen proteins, no signal. Two properly shaped proteins, you definitely get a signal, but one of each, you still get a signal enough to make the hair or the feathers grow down. So that's an example of a molecular mechanism behind a phenotype, so you can actually connect what these letters that we use to represent mean in a real life situation.